So I'm not going to do this today, but the anatomy of the glomeruli scream out to the observer. You have got to get a lot of liquid out of the capillaries here. In fact, over the course of a 24-hour period, about 180 liters of fluid will move across the glomeruli. Now, put that in context. We have about 5 liters of blood in our system. So to say the kidneys are efficient is a gross understatement. Okay, now, having mentioned the capillaries have to have special ways, let's talk about the 3 primary anatomies of those capillary beds that describe the importance of the simple squamous epithelial cells that form them. First, simple squamous epithelial cells in capillaries, in blood vessels, in places in your body are linked by protein junctions that act like zippers. And we call them tight junctions. Now, in these places where there are tight junctions between the simple squamous cells, you've got to have special transport mechanisms to move things across the capillary beds. Where in your body have you already learned that there are simple squamous epithelial cell linings that are joined by tight junctions? You learned this during the last class meeting. Where? Where do we find this? Sorry? No. There are desmosomes that join skin cells, but I'm thinking about blood vessels.
Your brain.
In your brain. So there are tight junctions between the cells in your brain. Now, before we leave that, let me just give you another memory tool there. The number one brain food, the thing that neurons like most, is sugar. What happens to your sugar levels during the course of your day? Do they stay constant? No, they don't stay constant. In fact, there are relatively dramatic ebbs and flows in our sugar levels. Now, certainly we have hormones to help us regulate that, but we do not want that kind of ebb and flow to have the kind of impact it could have on our brains. And so we need special transport mechanisms. You don't just let the sugar flow or not flow in the brain. There are special transport, facilitated diffusion mechanisms for moving sugar molecules across the tight junctions in the brain. What it means is you are a bit more steady neurologically than you would be otherwise. Now, does it have some impact? Sure, it has some. And if you want to see what that looks like, when sweet little Violet comes to visit you, give her a candy bar. And Dr. Payne will tell you later what she thinks about the tight junctions in the capillary beds of Violet's brain. Because she is going to get a surge, and that is going to cause a change in her behavior. Are y'all following me? Okay, so the tight junctions are important for helping to regulate, keep things more in balance. Second is fenestrated capillaries. Fenestrated capillaries are capillaries that have holes in them. For lack of a better illustration, it helps students remember, it looks like they've been shot with a shotgun. This is what the capillary bed looks like in the kidney. The kidney glomerulus is in fact a fenestrated capillary bed. So like I said, we'll come back to that again because I want to show you the pedicles there and the slit pores that are created by the connective tissues around those capillaries. This very important location. This is your first introduction, fenestrated capillary beds of the kidney. And then finally, discontinuous. I didn't spell that right, did I? Discontinuous. I'm not a good speller. Discontinuous capillaries are places where the capillary beds don't actually, the simple squamous cells don't actually meet each other, meaning they are discontinuous. And the best example of this is the capillary beds we find in the liver. So again, this is another very important place that we'll come to on more than one occasion in this class. Whenever material comes into the liver, it flows through channels. But those channels are created by cells that don't meet one another, which means that the entire tissue, liver tissue, is bathed in the blood that comes to it. You can see this in a dissection. In fact, in the cartoon pictures that you see of organs, when you're learning where the organs are in the various body regions, you will see that the liver has a different appearance to it. It's a deep, dark, red, almost purple color. Why does it look different? And the answer is because it is completely perfused with the blood flowing to it through the discontinuous capillaries, the sinusoids that are found there. Okay, a couple more. The blood vessels, I've mentioned to you, the lining of the heart, we'll do that with the endocardium when we get there. Very basically speaking, here, just a brief. As you move, so the anatomy of the heart we're going to learn, it's 3 big layers. But as you move from the chambers of the heart into the blood vessels, you have a continuous lining of epithelial cells. And so you will find simple squamous from the blood vessel continuing as a continuous sheet down into the chamber of the heart. So that's why we have lining of heart here with the blood vessels. The next one is lymphatic vessels. This is also going to be a very important anatomy piece in the last lab section, the last lecture section, the lymphatic vessels. Most students come to me knowing that they have arteries and veins. Most students come to me not knowing that there is another circulatory system in the human body, the lymphatic circuit, where there are capillaries and lymphatic vessels that are actually connected to the main circulation circuit of our bodies. Those guys have very similar anatomy. Guess what lines them? Simple squamous epithelium. And what would I expect to find in a lymphatic capillary versus a regular capillary? They're going to be the same. So all we'll have to learn when we start the lymphatic circuit for the capillary anatomy is, why do they stay open all the time? What's the special mechanism that the simple squamous cells use to form those swinging gates? How do you do that? So you've been introduced to it. Lymphatic capillaries are simple squamous epithelial cells. Their connections are unique. And then finally, the lining of the ventral body cavity, the serosa. Have you heard that word before? So if I wanted to open the body cavity and touch simple squamous epithelial cells, could you explain to me how you might do that? What is a serosa? Can you give me some examples of that? There are 3 primary serosal membranes in your body. Can you give me their names? The first one that we talked about was the pleura. The second one was the visceral and parietal. So pleura, and then around the heart we had pericardium, and in the gut we had peritoneum. So if I open up one of those bags, what is lining the serosa? Simple squamous epithelium. Okay. The next one on the list is simple cuboidal. You can see here for the simple cuboidal cells, they have tubes located here. And so I want to point out a couple of things in this slide that will be helpful to you. First of all, if you have a simple cuboidal layer, you mean one layer of cube-shaped cells. Okay, but when students look at this, sometimes they say, that's 2 layers. But that's not 2 layers. And the reason is, this is a section through the kidney tubes where the tubes themselves have been cross-sectioned. So if my finger was hollow, if it was a tube, what you're looking at in this picture is if I was pointing at you and I cut my finger this way. You're looking at a cross-section. So this white area in the middle here is the lumen the opening inside the tube. And this membrane here that lines the lumen has a name. It's called what's it called? The apical surface. Yeah. And what's the other side where the basement membrane, what's it going to be called? The basal surface or the basolateral surface. Yeah. So in this picture, lumen, apical surface, this is where the basement membrane is here, that's the basolateral surface. And you're thinking, wait, what? What? Is that really necessary for all of these? And the answer is yes. Because later in your lives, I'm going to have to teach you how it is you move things from the lumen inside this tube out into the interstitium so that it stays in your body. You know, if I'm going to push 180 liters of volume into these tubes every 24 hours, and I only have 5 liters of liquid in my blood vessels, then you can see that the kidneys would kill you by dehydration within hours were it not for the process of moving things out of the lumen back into circulation. And as it turns out, we know how that works. And so you're going to have to be familiar with these words. I'm going to start that conversation by saying, let me teach you about the proteins that are found in the apical membrane. Because these are the proteins that are going to be used to transport things across the lining of the tube, to get it out of the tube. Because if it stays in the tube, it's going to end up in the bladder. And I've got to get 178 liters a day out of these tubes. So I have to use these words, apical. Then we'll talk about all the proteins that are in the basolateral membrane. And you will learn the mechanism by which you get things out of the tubes. This is anatomy, introduction. You learn the apical, the basolateral, the cuboidal cells, the arrangement of these. Now, this is a picture of a very common picture of kidney. But you will learn from me, to point this out as an aside, an important aside, you will learn from me that the kidneys have other types of epithelial tissues lining things in them. Right? Can you give me an example? You start put these things together. Yes? Can you give me an example of another type of tissue we're going to find in the kidneys? The capillary beds there are fenestrated capillaries called the kidney glomeruli. Yes? Now we're also going to learn that some of the tubes in the kidneys are not cuboidal lined. They're actually simple squamous lined. And so that'll all be important for physiology. So learn the anatomy now. Cuboidal and squamous epithelial tissues of the kidneys. Now we also know that cuboidal cells are important for small glands, and they're found on the surface of the ovary, cuboidal epithelial tissues. Not as abundant, right? Not as abundant. Now, there is one more very famous place where you find simple cuboidal epithelial cells. And Mariam doesn't have it in the slide. I should have typed it in here for you. But I know Dr. Boyce is going to use it as one of her first examples. And that is in the thyroid. Thyroid tissue. In fact, at first glance, when I see this picture, my brain goes, thyroid. But then I see that the follicles are not taking up the right amount of space, and I go, okay, that must be the kidney. So thyroid tissue is also simple cuboidal. The follicles that make thyroid hormone, the cells that make the metabolism hormone in your body, they're simple cuboidal cells. Okay, so kidney and thyroid, and then not as important for me, at least not at this point, the secretory portions of small glands and the surface of the ovary. All right, the next one is simple columnar epithelium. And obviously, right, one layer column-shaped cells. Now, there's a bit more sophistication here for these guys because they exist in a couple of different varieties. The first variety to talk about is the non-ciliated type. Now, you already know as an anatomy student how to describe the anatomy of a cilium. Do you not? A cilium is constructed of protein in a 9+2 arrangement. And you have seen it already in this class as an illustration on pseudostratified columnar cells. Okay? This first mention of simple columnar is to say that there are in fact non-ciliated varieties of this, and this non-ciliated type lines most of the digestive system from the stomach to the anus. So now this is the second time I've mentioned this today. I mentioned to you as an illustration of the regenerative nature of epithelial cells. I said sometimes it's a problem when the stomach acid gets back into your esophagus because there's a transition in cells there. The epithelial lining of the esophagus changes as it moves into the stomach. Now you haven't seen the stratified squamous yet, but you've heard it. That transition is into specifically non-ciliated simple columnar epithelial cells. And they all have apical and basolateral surfaces. And you will also learn for cells in the stomach in physiology what kind of proteins on either side here doing all of the famous stuff of the stomach. You know, making acid and making enzymes and making mucus, and all those physiology things will be centered around the function of simple columnar epithelial cells. Now that brings up a point that I probably should have made already in terms of secretory things. Functionally, we don't normally think about squamous epithelial cells as producing products. Right? Did y'all hear that in my description? When I say simple squamous, your mind should immediately go to lining of blood vessels, lining of glomeruli. This is what we think about for simple squamous. Lining the serosa. Whenever I go to the cuboidal and columnar cells, I've mentioned to you the apical and basolateral surfaces as being functional things. And the only thing functional that I want to illustrate here is to point out to you, if you have a gland that makes something, that is producing some kind of product, you're going to expect that it's epithelial. And specifically, you're going to expect that it is either cuboidal or columnar epithelial. So when I see cubed and column-shaped cells, I think more sophistication than squamous, functionally, producing product. And obviously, this is going to be very important in the digestive system. All right. Now, the second type are the ciliated varieties, which line the small bronchi, the uterine tubes, and some regions of the uterus. All right, so let's take these in turn. First, the ciliated variety lines the small bronchi. Now, have you seen cilia already in my description of the respiratory system? You have. That was pseudostratified columnar. That's next. Yes? Now, from those two observations, you should already be putting together in your mind, oh, there must be transitions in the type of epithelium that I find lining the tubes in my body. So you should already start putting these kinds of things together. I'm going to do some more of this for you. But as we move through the respiratory system, we're going to see the types of epithelium there changing. And it's logical. It makes good sense.